SwayamPrabha. Digital India. Educated India.
Thank you all. So I'll briefly introduce myself as well. So I am a chemical engineer by training. I did my BTech in chemical engineering from IIT Durgapur, that's in West Bengal, and from that was in 2006 to 2010. And then I went from BTech, I went directly for my PhD. That was also in chemical engineering, and that was in USA in a state called Alabama, Auburn University, and I was there for about 6 years. And then from there, I went to another state called Delaware for my postdoc. So I spent 3 years there. So I was there in the US for about 9 years or so. And then I returned back and joined here in IIT Madras as a professor. Okay. So I am primarily a chemical engineer. I'm not a biologist. Okay. So I do whatever biology I know is up to class 11, class 12, whatever we learn. And after that, I started learning about chemical engineering. So I shifted away from biology. And then gradually, as I was doing my PhD, I started learning a little bit more about the biological world. And then as part of my research, and then I came back here and started joined here in biotechnology department. Okay. So if you ask me a very deep biological question, I may not be able to answer. But I will try my best to answer. Okay. So I work in the area of material science little bit and a little bit of biology, and I try to merge these two fields. And one of the important areas that I work or tools that I use in my work is fluorescence microscopy. Okay. So that is what I am going to talk to you about. Okay. So if you have any questions, feel free to stop me anytime. Raise your hands and don't be shy. Try and don't worry about what questions or anything. Any type of question you have, you just ask me. Okay, I'll try my best to answer. Okay. Just to start, the world of microscopy. Okay. So you know what is a microscope? What is a microscope? Right, right. So the world of microscopy actually did not start from microscopy, it started from astronomy. Okay. So as you know, astronomy is a very, very old field. There are Indian astronomers, Chinese astronomers, Egyptian astronomers who have been observing the sky for many centuries, long before. So they have been working on telescopes, how to develop better telescopes to see even further and further things. Okay. So the first person that you see on the left is Galileo. Okay. He is known as the father of modern astronomy. Okay. Can you name some of the things he discovered? Huh? Okay. Related to astronomy? Yes. What did he discover with the telescope? Saturn. Correct, yeah. So he discovered things like Jupiter's moons, rings of Saturn, the phases of Venus, the sunspots on the sun. So he developed all of these and he did it with his own telescope. Okay. And so not just him, there were many other parallel astronomers who were developing these telescopes. And so they ushered in the field of modern astronomy. Okay. And that time Europe was going through a period called Renaissance. Okay, it was coming out of its dark ages. A lot of new scientific discoveries were happening. So Galileo and his fellow scientists, they were all part of that new Renaissance. Okay, and they were making a lot of discoveries at that time. Now in a telescope, how does it work? What is there in a telescope? It's a long tube, right? What is there inside the tube? Lens. Good. So there is a convex lens. Okay. So this is also kind of a lens, right? And lens bend and diffract light, right? So by manipulating these type of lenses, the scientists were able to see very far away objects with their human eye. Okay. So now, during that time, during and after Galileo, a guy named Leeuwenhoek, okay, he was a fellow, he was not a scientist, okay, he was just a common person. He used to fiddle with these lens. So in Denmark and Germany and all, there were a lot of people who were grinding these lenses for telescope application, for binocular application and all that. Okay. So this guy Leeuwenhoek, he had an idea that instead of pointing this lens at the sky, what if I point it at the ground, okay, in the soil or in the water. Okay. So what he did, he took a lens like this and he took a drop of rainwater. Okay. He observed that rainwater through this lens, and he's noticed something amazing. He noticed things which are swimming, some things were circular in shape, something were rod-like in shape, something had tails in them, something had hair-like brush around them. Okay, so he was very surprised. What are these things? We can't see it with our normal eyes, but when I see it under the lens, what are all these things swimming in the water? And he saw it in the rainwater. He collected water from the pond. He saw it there also. He collected water from various sources. Wherever he saw these swimming things. Okay, and so he started making drawings of these things. Okay, so some long, some short, some circle, all of these drawings. So that time there was no camera. Camera had not been invented that time. So all he could do was make these drawings. Okay, and then later on, so he is considered the father of microbiology. Okay, so later on, the people who came after him, they were able to say that these are actually some living things which are of the micron scale or even smaller. Okay, and these can be later on people like Koch and others, they classified them into bacteria, protozoa and other things like that. Okay, and so he is known as the father of microbiology because he first observed these things and made detailed drawings of all of this. Okay, so this is how the world of microscopy first started. People came to know that there is a whole world which is not visible in our normal eyes, which is very small, microscopic or even nanoscopic in nature, which can be observed through that. Okay, so he developed this, and the kind of microscope that he used is like this. It is about this size. Okay, and you can hold it in your hand like this. Okay, just like you hold a pen, you can hold it like this, and you put your drop of water here. The convex lens is here. Okay, this very dot. You just put a drop of water and then you hold it with your eye. Okay, like this, and you can see those things in the eye. So this is kept in a museum in Denmark, the one which he used for making all of these discoveries. Okay, so that is how the world of microscopy started. So now with this, as you can see, this was like what in the 1600, 1700. So then people got more and more fascinated. What are these things? How many of these things can we see? How many different shapes are there and all that? So now, after decades and decades of innovation and discovery were made on this, people started identifying which bacteria causes which disease, which protozoa causes which disease, which one is found in which type of water, how do these, what are the structures of these, what is there inside them, what is there outside of them, what does this tail do, what does the cilia on the surface do, all of these things. Okay, so people made a lot of discoveries on that. So coming a bit later, this guy in India, that time British were ruling India, a lot of British officers in India were falling sick of an unknown disease. Okay, they were getting a high fever and they were dying a lot. Okay, and people could not understand why they were dying. They were giving some medicines to cool down the fever, but still people were dying. Mostly in the tea estates of Assam, West Bengal, Nilgiri Hills and all that. So people were, all the British officers were wondering, what is this disease? Why are people dying? This is not like some stomach bug or some dysentery or typhoid or something. This is some unknown disease. And they could not identify what is this disease. So this guy, he was originally trained in England. He came here as a British officer. He set up his lab here in a hospital in the Nilgiri, in the Uti region. And there he started thinking about what could be causing the disease. Okay, so he collected some water from the wells, and he tried to look at it under the microscope. He could not find anything that would that is unknown. He saw many things, but nothing that causes that unknown disease. Okay, so then he had an idea that this disease is caused only where there is a lot of mosquitoes. Okay, and he thought, is the mosquito somehow related to this disease? So then he started collecting mosquitoes from different regions. Okay, and he started dissecting the mosquitoes under the microscope and started looking at them. What is there? He cut the legs, he cut the stomach, the intestine, the salivary gland, and everything. So there he started noticing that in the stomach there are certain round objects. Okay, and he started making drawings of these objects. Okay, and then in some species of mosquitoes he found them, in some he did not. So he was also confused what is happening. And then he also found out that he dissected the salivary gland of the mosquito. Okay, and so there he found that these kind of things are there. So then he found that this, named it as Plasmodium parasite, and he found this parasite that coexists with mosquito, and then as the mosquito is sucking the blood, this parasite gets transmitted from the salivary gland of the mosquito to the blood. Okay, so it happens in humans and it happens in birds also. Okay, so he primarily showed that it happens in birds. Another guy showed it in humans. Okay, so he was the first person outside Europe to get the Nobel Prize for discovering this Plasmodium parasite, and he did all this with a microscope. Okay, so at that time there was no camera. He had to draw all of this by himself. He had to write down detailed notes like mosquito number 1, I found this thing. Mosquito number 2, this thing was swimming. He had to write notes like this. Okay, and he had to draw detailed drawing. Okay, otherwise people will not believe what he saw. He cannot go around with his microscope showing it to people, right? So he has to make detailed drawings of all his notes. So all of sorry, all of his notes are there in the Nobel Prize museum. So you can read up his notes. It's a 100-page note filled with all kinds of detailed drawings and recordings of his observation. And sometimes he wrote things like, so that time there was no light, there was no bulb. Okay, what people used to do, they used to light the candle, and with the candle, light of the candle, they used to observe these small things. Okay, and then in India that time there was not much research infrastructure also, so he had to somehow assemble his microscope. And so if you read his notes, he will say sometimes he is very frustrated, like I'm sweating all day, my fan is not running, the sweat from my forehead is falling on my sample, I am not able to work properly, I don't have a good microscope. So he has written all these notes in him. So you know how much he had to struggle to make this discovery. Okay. So that was the story of this discovery. So as you know, this kind of microscopy technology actually ushered in a lot of discoveries like this. Okay. Since that time, this microscopy field has been making a huge revolution. So you can see many different things. You can see human diseases, you can see plant cells, microbial cells, blood vessels, many kinds of things, all kinds of microorganisms you can see. And different kinds of techniques have been developed. Okay. I am not going into the details of all of this. You can note down the names and you can look it up on Wikipedia. So the way the light passes, you can see certain things inside an object. The way, if you bend the light in certain way, you can see the light, the things that are surrounding that object. Okay. So for example, if you see our human cells, okay, you know that our human cells are filled with water, right? There is how much percent water in our body? 70%. Now water is transparent, right? So if you look at a water filled object inside the microscope, what will you see? Nothing, right? Blank. It's transparent, all the light will go through. So then how do we see the things inside our cell? Huh? Staining is one way, yes. The trick is to make the light bend around things. Okay. Diffraction. Diffraction is one way. Another way is called phase contrast. Okay. So if you split a light beam and make it pass through and diffract and bend around certain things, then you can see solid objects within a transparent view. Okay. So for example, if you look at this image, okay, so you can see that there is 1, 2, 3, 4, 5 cells. Okay. There are 5 cells. These are some kind of human cells. Okay. This dark object that you see in the center of this is the nucleus. Okay. And the surrounding region that you see around them, that is lighter in color, that is the cytoplasm. Okay. And so the nucleus, if you just look at it under the blank light, you will not see anything. You will just see, because it is 70% water, you will just see some dark spots. You will not be able to see all these finer features. So because the light is getting bent in a certain way, so you can see each cell is surrounded by a bright ring of light. Okay. If you see my laser pointer, each cell is surrounded by a bright, slightly glowing kind of light. Okay. So that is the feature of phase contrast. Okay. Phase contrast is a way where the light beam is split in multiple beams. It passes through an object, and then these multiple beams, because it is passing through lighter region, denser region, then it gets recombined again and it gives you this kind of glowing feature. Okay. So then you know where is the boundary of each cell, where is the boundary of the nucleus, which part of the nucleus, what is there inside the nucleus. There are some dark regions in the nucleus which are the chromosomes. There are some lighter regions in the nucleus. And then within the cytoplasm also, if you go higher, you look closely, you will see certain darker regions and all that. Okay. So that is one good feature of phase contrast. Okay. So people have developed this kind of technique. There are other kinds of techniques also. So if you invert this image, here you see a light background and a darker object. You can flip this. Okay. You can have a dark background and a lighter object. Okay. So that is called dark field microscopy. So there, if you are having let's say some microscopic organism whose internal structure you want to study. Okay. So if you can make that background dark and the internal structure starts glowing, so it will become more prominent in your eyes. Okay. So you can start seeing its food pipe, and other internal organs, stomach and all those things that are there inside it. Okay. Now some people may be interested in the surface things. They may not be interested in the internal thing, they may be interested in the surface, whether the surface is smooth, whether it is rough, or whether it is bumpy, or what it is. So there is another kind of microscope which people use, and that is called this DIC. Okay. So in this DIC, this is also a human cell. Okay. So this is one cell, this is another cell, this is another cell, this is another cell. Okay. But here you cannot see the inside of the cell. Okay. Here you are seeing more on the outside, the outer surface. So the bumpy part that you see in each of them, that bumpy part is the nucleus. The nucleus is a elongated like a egg shaped shape, and that gives this bumpy kind of appearance to the cell. So if you look under DIC, that is called differential interference contrast. So if you use that kind of microscope, you will see the bumpiness of the nucleus, but you will also see the outside surface of the cell, how it is stretching, how it is all these fiber like things that are there in the cell. Okay. So all of these things become prominent. Okay. So this is one way of people observe things. Another one which one of you said was staining. So right now we are seeing only in black, white and grey, right? But people may want to use some colors to see whether there is a difference in the way, whether we can distinguish objects from one another. Okay. So the problem appears is, let's say let's say I'm looking at the sky, okay. Let's say there are clouds in the sky, okay. Now let's say some clouds are big, some clouds are small, okay. Let's say I want to observe a small cloud, okay. How do I find that small cloud, whether it is there in the sky or not? Hmm?
The big clouds go in there, you see the small cloud.
Hmm.
So hence, you can't technically look at that, but then you can go high and then see the small cloud.
What would happen if the cloud is also blue in color?
Oh, then you can see the small clouds.
The sky is blue in color and the clouds are also blue in colors.
You can find a way to make boundary shades color. You can see the boundary smaller and
Okay.
Instead of looking at the clouds themselves, you look at how much light is scattered by whatever is coming through. So you can see that there's a cloud and light is scattered more as opposed to here, like this sky. I mean, if you have a big cloud or a small cloud and it's happening in that region, more than just scattering light than just a big.
Yeah, you can do that. You can see the scattering of light, like how heat here happens here, right? Here the background is also gray and your object is also gray, right? But still you are seeing, you are able to distinguish the object from the background. But so if you are seeing the clouds, let's say your clouds and your background, the sky is of the same color, you can apply that kind of technique. But another kind of technique is to make it of a different color, okay. So that is what is obtained with staining, okay. So in staining what happens, if there are certain dyes, okay. So those dyes will stain the background in one color and your actual object in another color, okay. So let's say here, this is some kind of tissue of the human body, okay. So there are many cells here. So each of these violet dark spots that you see, that is a single nucleus, okay. So here they have used a stain called hematoxylin and eosin. These two stains, one stains the background in a light purple color and another stains the nucleus in a dark purple color, okay. And then there are some parts which are not stained at all, so they will appear white, okay. So then you can distinguish which part has a high density of cells, which part has a low density of cells, and which part is filled with some kind of fluid, okay, like water or some other kind of fluid, okay. So if you look at a tissue sample under the microscope, you can distinguish whether there is a high density of cells, whether there is low density, or whether this is, let's say if you have some cancer growing in the tissue, okay. Cancer is a situation where the cells are dividing rapidly, okay. So the cells will be jam-packed with each other. So if you look at a cancer tissue under the microscope, you will see a certain region where there is a lot of densely packed purple spots, okay. So then the doctor can know that, okay, this part has some kind of tumor growing, okay, which is different from the normal healthy tissue where there is more uniform distribution, and there they can identify that, okay, this is a problem, okay. So that kind of staining helps you to identify irregular patterns or distinguish between normal and healthy diseased tissues, okay. Any questions? All right. Okay. So I'm just giving you the basics. I think many of you already know it. So either in telescope or microscope, the central principle is having a objective or having a convex lens, okay. So convex lens, depending upon the radius of curvature, you can get different kinds of magnification. In some convex lens which have low radius of curvature, you can get magnification of low amount, okay. And all of these have something called refractive index. I'll come to that. So if you have a small object, okay, the light as it passes through this plane, it gets bent, okay, and different parts of the light as it passes through different parts of the lens, it gets bent in different degrees, and then it goes into the human eye, and the human eye back projects it like that to see the magnified view of the object, okay. Right, right.
So, how do we correct for that in a microscope?
Huh. In microscope also, I'll show you in a bit. There is a series of convex lens, concave lenses, which are able to subtract out the aberrations. So it's a compensating feature. I'll show it to you in a bit here. So the basic principle is like this: you have a convex lens, and the light coming to your eye gets magnified. Now, if based on the degree of curvature of this convex lens, you can get bigger and bigger objects. Okay. Now, this is another concept. I think you already know it. You have two mediums, let's say air or water. The water you can think of it as glass also. Okay, the glass of the convex lens. So, as the light travels from one plane to another plane, it gets bent, and the angle that it makes is called the incident angle and the refracted angle. And depending upon the angle at which light is passing, at a certain angle it will pass through, and at a critical angle it will it will follow this plane. It will not go into this plane at all. And if you increase the angle of incidence, it will get totally internally reflected. Okay, so that's called total internal reflection. So these principles, the total internal reflection and this refraction, is used in the principle of microscopy. Primarily, this principle is used in microscopy, but I will also show you later about total internal reflection, how this is also used in microscopy. Okay. So the basic principle is again same thing, going back. The light is coming here, passing through your convex lens, and depending upon the radius of curvature, you can magnify it to 4 times, 10 times, 20 times, all the way up to 100 times. Okay. So there once if you see any microscope, if you look at the objective, that is your convex lens, there it will say, it will be printed there how many magnifications, how many x. So you will either see 4x, 10x, 20x, 40x, or 60x or 100x, something will be printed on the side. Okay. So using that, you will know what is the magnification of that particular objective. Okay. Yeah. And then there is this concept of numerical aperture. Okay. So I'm just going through a little bit of the physics, and then I'll go into the actual microscope a bit later. Okay. So in a microscope, you will have a tube like this. Okay. That tube will have this kind of lens in it, and then you will put your sample here, whether it is some microbe or cell or something. You will put your sample here. There will be a light source which will shine a light, and this light will pass through your sample, and then it will make an angle through your sample, and this light will again pass through, and your eye will be on this side. Okay. Now, depending upon this angle, you can so each of this objective will have a certain angle. So you will have to focus your objective. So let's say like how we do with binoculars, right? We either we try to focus it so that we can see a certain distance. In a microscope, what we can do is we can move the lens closer or farther away from our sample. So depending upon this angle, we have to decide whether we need to go closer and get in the line of plane. Okay. Any questions so far? Now, the thing that you said, diffraction. So the problem that happens in many microscopes is that when you are having a point source of light, let's say I want to see a single point of a point object. Okay. Now, when I try to see it under the microscope, due to the light bending under this, I will not see a single point, but I will see a pattern like this, a ring-like pattern. Okay. So this is called a airy disk pattern. It was first observed by Newton, and he explained why this happens. This pattern, so if you have a single point source of light, this will appear like a ring-like pattern here. The brightest intensity of light will be here, which is about here, 84%, and then the surrounding peaks will be having a ring, gradually decreasing ring-like pattern here. Okay. Now, the problem is, this is a big problem. So, if we want to see, let's say, one micrometer small object, okay. Now, let's say I want my object is circular in shape, okay. But now I am seeing this kind of ring-like shape. So, I may be fooled, right, that why is this ring-like shape happening? Is my object like this? Or let's say I'm observing an object that is this shape, okay. Now, if I want to see it under the microscope, this Newton, because of this airy disc, I may start seeing this kind of artifacts in my eye, okay. So, this is a big problem, and so there are ways to correct for this, and one way to correct is what you call correction of the spherical aberration or even chromatic aberration. So, what we do is we place a series of objectives or lenses in plane so that they subtract out each other's distorting effects. Okay. So, there are two kinds of aberrations or two kinds of distortions. One is called the chromatic aberration, that is distortion of the color. Okay. So, as you know, white light is made up of different colors of light according to the, you know the colors, right? The fluorescence spectrum. So, now different colors of light will bend through the objective to different degrees. Okay. So, sometimes green will separate out from the red or blue will separate out from the green. Okay. So, that is one kind of aberration, that is called chromatic aberration. Okay. There is a way for correcting that chromatic aberration, and then there is another called spherical aberration. Okay. So, this is this airy disc pattern is one example of the spherical aberration. So, even if your object is a tiny dot, it will appear like a ring-like airy disc because of that aberration. So, there are some ways of correcting that aberration as well. Okay. And when you are observing this, the kind of pattern that is formed, that is called a point spread function. Okay. So, I am not going too deep into the physics of it, it is a bit higher level physics, but you can just note down the term point spread function, and people try to make their lens in such a way that this central peak is as high as possible and the surrounding peaks are as low as possible. Okay. So, that it appears like a single peak of light or single pinpoint of light. Okay. Okay. So, the reason that this problem is happening is because of this. Okay. So, let us say I have some bacteria that are swimming in the drop of water in my sample. Okay. Let us say this is my bacterium. So, those who are sitting in the last benches, can you count how many bacteria are here? Huh? First bench, okay. So, what is the issue? Why you can't count? Close to each other. So, if you come here, can you count? Huh? Right, overlapping. So, if they are overlapping with each other, how do you know whether it is one or is it two? But how do you know whether it is 2 or 3 or 4? So, that is where the issue of resolution comes. Can you resolve 2 points in your field of view? Okay. So, when you are looking at 2 things, let us say if you are looking at the sky and let us say there are clouds in the sky. Let us say 2 different clouds are very close to each other. Can you see with your eyes whether they are two separate or whether it's a single cloud? It depends on the resolving power of your eye. If your eye has a certain resolution power, okay, if you with that you can identify to some extent whether if two small clouds are separate from each other or whether they are part of the same big cloud. Okay. Now, if you want to go better than that, then with a telescope or binocular, you can look at it at the same cloud, and there with that maybe you can distinguish whether these two clouds are actually separate or whether they are close to each other. Okay. So, that is called the resolution limit. Okay. So, that is the issue that happens in the microscopic world also. Okay. So, when two points are very close to each other, okay. So, for example, this one here, the image that you are getting, we cannot say whether this is one dot or whether it is two. Okay. But the ones which are far away from each other, we can definitely say that they are distinct. Okay. So, the ones which are close to each other, we can kind of say, so that is the resolution limit. Okay. So, the power of your microscope or the objective, or sorry, the lens that you have lies in this, whether it can resolve these two objects from each other or not. Okay. The better your resolution limit, the better is your microscope. Okay. And that is dependent on this formula. This is called Abbe's equation. So, Abbe was a great physicist who did a lot of physics, and he arrived at this final formula. This formula is so important at that time that in his gravestone this formula is embedded. Okay. So, you can look up this guy's name, Abbe. So, the entire world of microscopy is dependent on this formula. Okay. And so, he said that the resolution, the limit of this resolution is lambda, that is the wavelength of light, divided by 2 times n, that is the numerical, that is the refractive index, multiplied by sine of alpha. Okay. The alpha is the angle that is made by this lens here. Okay. So, if you know the angle alpha and you know the refractive index of your lens, of your glass. So, this glass can be made up of quartz glass or silica glass or fluoride glass. Different glasses have different kinds of refractive index. So, based on this you can get this resolution limit here. Okay. And depending on the wavelength of light, you can plug it in this formula and you will get whether you need a red light or whether you need blue light or whether you need green light to resolve these different points. Okay. And so, a typical microscope will have multiple objectives like this to correct for compensate for the resolution or the aberrations that happen. So, your your actual sample will be here. Okay. Your light will come from the bottom and there will be a part called condenser. The job of the condenser is to condense that light, infinite beam of light into a point source so that it falls on your specimen, that is a sample, and the light that passes through your sample again goes through a series of lenses and then it goes to your eye or the camera. Okay. So, this whole process gives you this kind of magnified image. Okay. Shall we take a break now? Okay. After 5 minutes, okay. Two. Okay, 10 o'clock. Okay. All right. So, there are different kinds of objectives. Okay. So, some objectives are give you a magnification of 4 times, some objectives give you a magnification of 100 times. Okay. So, you need to know which type of objective you want and what is your sample. Okay. So, let us say I want to take a image of this let's say this water bottle, okay, imagine this is my sample. I want to take image of from the top to the bottom, this whole thing, okay. Now I place it on the microscope stage, okay, and because the height of this is very big, or the size of this is very big, I cannot use high objective lens, okay, because every lens has a certain working distance, okay. So every lens has a working distance, and that working distance is determined by this, okay, by the angle or the numerical aperture. So let's say you have a objective which magnifies 4x times, okay. Now this 4x, your sample is sitting here, you have a big sample, thick sample, it is sitting here, your 4x object lens is here, okay. Your light is making a very small angle as it is going from the sample all the way to the lens, okay. Now because your object is, because this angle is small, so your 4x objective can be very far away from your sample, and you can capture a lot of things in, if you have a thick sample in this region, okay. So that is one advantage of this system. But in a 4x objective, you can magnify only 4 times, okay. Maybe in that 4 times magnification, you may not be able to capture smaller things, you may miss out on that. So what to do? Okay, change the magnification power. What did you say?
Increase the 4x like 5 or 6.
Huh. Okay, same thing, yeah. So if you increase that magnification, let's say instead of a 4x lens, I use a 100x lens, okay. So now the problem with the 100x lens is this angle is very small, is very large, okay. So the light that is coming from your specimen has to make a large bend, and your objective or your lens has to be really close to your sample, okay. That means if I want to image a big sample, I can't do it, okay. So what I have to do, I have to slice my sample, make very thin slice, and each slice I have to put it under this in the holder, okay, and then I can use the 100x objective to see the slice by slice view of my whole object, okay. So that is the problem with 100x. You will get a larger magnification, but your working distance will go really down, okay. So you have to choose whenever you are given the option. Let's say you are given a microscope, okay. First thing you notice is what is the magnification that is printed on the label. If your magnification is low, that means even from a further distance you can see the sample. But if it is a higher magnification like 40x, 60x, 100x, then you need to be very close to your sample, and your sample has to be very thin, like a thin smear of blood or a thin film of some material, and only then you can see at the 100x, okay. So that is, so you have to make a trade-off here, okay.
SwayamPrabha, Digital India, Educated India.